Thank you so much for joining us today for the e-seminar series on translational biomedical engineering. This is our 83rd seminar since June 2020. So it's our great honor to have our friend and colleague, Professor Ali Tamayol from Department of Biomedical Engineering, University of Connecticut Health Center, that is talking about translational biomaterials engineering system for treatment of skin and musculoskeletal injuries. So before going to his talk, if you have any question, you can put your question in the ask a question box on the bottom of your screen. And for the chat box also, you can have the, formal chat. You can say, where you are coming from, or, chat with other attendees. So if you want to get the most updated information for the upcoming seminar series, you can follow us on Twitter. Our account is hashtag, handle TransLBME on Twitter, or the organizer of this seminar, Professor Akbari, or myself on LinkedIn, so to get the most updated information of the upcoming seminar. So our next speaker next Wednesday at noon Eastern Time is Professor Dino Di Carlo from Department of Bioengineering from the UCLA. Last but not least, I would like to thank our sponsor, Montreal Transmedic Institute, which sponsors this seminar series. By that, I will introduce our speaker today. Professor Tamayol is an associate professor of biomedical engineering at the University of Connecticut Health Center. Prior to this appointment, he was an assistant professor at the Department of Mechanical and Materials Engineering at the University of Nebraska, Lincoln, and an instructor of the medicine at Harvard Medical School. He received his bachelor degree from Shiraz University, followed by a master's from Sharif University of Technology in Mechanical Engineering. He received his PhD from Simon Fraser University in Canada in 2011. He did 1 year of postdoctoral fellowship in biomedical engineering at McGill University and 3 years of postdoctoral fellowship at Harvard Medical School and Massachusetts Institute of Technology, MIT. His research involves design, fabrication, and characterization of microsystems and fibrous materials for emerging engineering applications such as tissue engineering, regenerative medicine, and wearable devices. He has authored more than 150 journal papers, 5 book chapters, and 14 patents. In addition, he has given over 50 seminars at various conferences and academic institutions. His publications have been cited more than 11,000 times, and he has been the recipient of several awards, including NSERC Postdoctoral Fellowship, BCIC Scholar Awards, and the Ali NASA Prize of ISME. And this list goes on and on. By that, I would like to invite our speaker to share his screen and start his talk. Thank you so much, Professor Tamayol.
Thank you so much for the invitation. It's a pleasure to be here. First of all, as you mentioned, we have been friends and then colleagues, and I really appreciate this opportunity. So, as we discussed before, the theme of the kind of this seminar series is more on the translational research. So I try to kind of focus on the translational aspect of research that's being done in my lab. But overall, in my lab, the research has my lab research has 2 pillars. One is treatment of skin injuries. The other one is focused on treatment of musculoskeletal injuries. And today I try to touch some of the recent progress that we made in these areas that I feel that they're translational. So I'm going to start with the area of musculoskeletal tissue engineering, or treatment of musculoskeletal injuries. So perhaps you guys have heard before that musculoskeletal injuries can be caused by traumatic events, tumor removal, chronic diseases. the numbers are staggering, right? Every year, about 1 million cases of bone grafts are being done in the U.S. 500,000 patients are suffering from volumetric muscle loss, and usually these patients are going through multiple rounds of surgical procedures. So this is basically a major challenge. Although the musculoskeletal tissues are known to have or possess excellent regenerative capacity, but underlying conditions, extent of the injury can overwhelm or overpower these regenerative capacity. But then today I'm going to mostly focus on volumetric muscle loss, but some of these technologies that are introduced can be translated, or we have applied them for treatment of bone defects, tendon injuries, etc. So when it gets to treatment of volumetric muscle loss, the gold standard, there's no gold standard based on my discussions with clinicians. Right now they use muscle flaps, but it's not very common, and also usually they don't lead to functional recovery. But then when clinical practices fail, we turn into alternative strategies, and the most exciting alternative strategy, or the most promising alternative strategy, is tissue engineering. So, the goals in tissue engineering is first create an environment that supports cellular growth, creating basically an environment that direct organization of myotubes, because the organization and the anisotropy of the myotubes is key to their function, and then tailoring mechanical properties to achieve biomechanical properties both at the cell level and also at the tissue level. And then obviously, when you have tissue engineering efforts, you should think about its connection to the host body through proper innervation and proper vascularization. So, but VML typically lead to severe fibrosis. It doesn't regenerate. And when we are talking about volumetric muscle loss, we are talking about the case that 20% or more than 20% of mass of muscle is being removed, a single muscle. So then usually it leads to severe fibrosis. It doesn't regenerate much. It leads to permanent loss of function. And there are different basically some hypotheses for this scenario, including the lack of regeneration, lack of mobility of satellite cells or progenitor cells that are supposed to help with the regeneration, inflammation that helps or directs the body to more of reparative systems like fibrosis rather than regenerative mechanisms. But then there are also some other things that are involved when you're thinking about the treatments or tissue engineering strategies, which is the logistic of these strategies. How, when you fabricate something, how to do the implantation, etc. So just to give you an idea about the typical VML injury model and what we see in that, this is a model that we generated in collaboration with my colleague at Brigham Women's Hospital, Dr. Indraneet Sinha, and we basically dissected a portion of GA muscle, and then after 8 weeks, when you go back and look at the muscle, you see the difference between the basically architecture of the healthy population and non-treated VML injured muscle. And as you see, there is a significant functional deficit in the animals post-injury. But one of the interesting points that I want to mention is that another cause of these lack of regeneration is the significant change in the level of cytokines and biological factors that are supposed to contribute to the regeneration. For example, one of the important ones is IGF-1, which IGF-1 is we have seen that post VML injury is being significantly downregulated, and IGF-1 is actually an important factor which contributes to immune modulation. It supports proliferation of progenitor cells as well as their differentiation downward. So, imagine a piece of muscle is gone, the extracellular matrix is gone. The cells that are responsible for regeneration are not also, basically available or effective, right? And the factors that are needed to mobilize them and direct their function are also not available. So we have developed many different strategies, just different strategies. We work with Dr. Akbari on utilizing textile processes and in the field of utilizing textile tissue engineering for making muscle construct and moving forward for implantation, which wasn't very successful because the architecture of the textile structures that we fabricated typically are not very compatible with the types of injuries or the architecture of the injuries that we are dealing with. Then we also did 3D printing and bioprinting, because when you're dealing with complex architectures, that's one of the most exciting strategies, because you can tailor the architecture easily. But then one of the issues that we faced is that if you are dealing with hydrogel constructs, the implantation is not trivial, right? So then you either have to glue them, or you have to find other strategies to hold them in place and avoid their dislocation, which is not again very convenient. But the other thing is that typically some of these injuries occur because of traumatic injuries or traumatic events, or during surgical procedures, right? So then you can plan ahead, right? So then you need things or tools to be able to utilize or deploy during surgery. So with what they call intraoperative tools, right? So then to be able to get there, we try to look into or try to modify things and make things portable, or some sort of point of care printing tools, in a way that they can be realistically deliver the material or during the surgical procedure. But also we design it in a way that there is no need for being worried about the adhesion or the implantation and dislocation of the scaffolds down the road. So what we developed was a very simple extrusion device, which was a handheld kind of printer, and the material was loaded into the syringe, and there was a UV light attached to the system. We used gelatin methacrylate. We delivered the material and then cross-linked the material in situ. The good thing about utilizing gelatin methacrylate and cross-linking or in situ cross-linking is that once you cross-link material directly on the tissue or at the interface with tissue, there is a reaction between methacrylate groups on the material and also the amine groups on the tissue. So then there is some sort of hydrophobic or basically interaction between the materials, and then that leads to adhesion, proper adhesion. So the material is going to adhere and stay in place without using any suturing, without the need for using suturing. But at the same time, you can utilize this system for delivery of cells, and because of the shear stress that's being induced during the extrusion of the material, that is going to direct the organization of the cells and topography and organ alignment of the cells. But initially we thought, maybe inducing vascularization early on is going to be an important factor, and so we try to make or incorporate VEGF, which is an important biological factor inducing angiogenesis, right, into the scaffold, and then follow the same trend, right? And for control releasing the factor, we decided to utilize laponite, which is basically nanoclay material, commercially available nanoclay material, and it's an interesting material. It has positive and negative charges, so you can utilize it for control release of different molecules with different charges. So we managed to control the binding of protein. We looked into the release kinetic. It's a very gradual release kinetic, started with relatively a bit of kind of burst release, not significant burst, but it's going to be gradual. But then incorporation of laponite moderately affect the structure of the gelatin methacrylate, and one of the things that we noticed actually makes the pores larger. But at the same time, the laponite is a hard material, so then overall mechanical properties did not change at the levels that we were incorporating the material. But then we also looked into the adhesion, and as you can see here, the adhesion also was not significantly impacted by the presence of the laponite. So we created basically a volumetric muscle loss injury in mice, and then as you can see, this pen printer can be easily utilized to rebuild the architecture of the lost muscle. And then after 4 weeks, we went and basically ran the animal and to see a potential functional recovery. The results initially suggested that, well, the right scaffold, the scaffold that carried VEGF, contributed to some level of functional recovery in terms of, improving the maximum running speed and maximum running distance of the animals. For maximum running speed, there was no statistically significant difference between the healthy population, but for endurance, there was a significant difference. But then when we went and performed the histological analysis to see what's going on, and if we have tissue regeneration, the only thing that we noticed was that we have less fibrosis, and this is the trichrome staining, and blue is showing fibrotic tissue. So then we didn't have fibrosis, but we didn't also have significant regeneration. But when we looked into the remnant tissue, we noticed that basically the size of the fibers, muscle fibers in the remnant tissue has increased. So then it's the condition that is referred to as hypertrophy. So the hypertrophical changes in the muscle contributed to the functional recovery. But then the question that we had was why we didn't see regeneration. The material gelma is known to be supportive of cell growth, cell differentiation, etc. in vitro. Why it didn't generate any regeneration in vivo? So then the assumption that we had was that gelma is a dense material, and then if you expect the cells to migrate into that, then this means that cells have to utilize energy to be able to get to degrade the material, remodel the material, basically open pores for themselves, start proliferating in the material. So probably it's too much to ask from these cells, right? So then we decided to generate some larger pores in the materials, and to do that, we start to look into some of the things that we have around us, like whipped creams, right, or egg whites that if you basically agitate them, mechanically agitate them, they're going to generate foams, stable foams. Also, thinking about shaving foams, right? When you press the canister, you see a very interesting foam is coming out, and the foam is actually very stable. So the good thing about foam is when you have foam, it's actually easy to print. It's very different from gelatin or gelma inks. So it's actually behaving as shear thinning, although it's not a shear thinning material, but the behavior is kind of similar. So when you extrude it, it's going to go out, but still it's going to maintain structure. But when we looked into the architecture or microarchitecture of gelma foams, you can see that you see different levels of porosity, right, ranging from the pores that are produced using 3D printing or bioprinting to macropores that are generated by the air bubbles that are being trapped, and then nanopores or very small pores that are inherent to or part of gelma characteristics, right? So then we had larger pores connected to each other through nanopores, right? So that was the architecture that we saw. It was a hierarchical porosity. And then we did some 3D printing to test if the material is easy to print. We could fabricate structures with relatively good, resolution. It could form three-dimensional structures, and even it could form freestanding structure, which I don't have it here. But the other good thing is that these foams, if you mix them with different proteins or different molecules, the diffusion is very slow. It's much slower than gelatin methacrylate on its own, or gelatin methacrylate gel on its own. So then you can easily create basic distinct regions, and even if you want to generate some gradients, you can easily control and make them. But then we started to utilize this system for treatment of the animal model that I showed before, and this is actually a very interesting picture that I'm very proud of that when I share. And this is, in here you see 4 animal groups. And again, I should probably step back. So in this experiment, in addition to going with just foam on its own, but also we tried to look into if we can utilize foam for control use of IGF-1, which is a factor that we knew was being downregulated. So we had 4 groups. One was sham injury, the other one was VML injury, which was left untreated. We had another group which was VML injury, which was treated with base foam, and VML injury, which was treated with foam plus IGF-1. So then as you see here, both cases that receive the scaffold, you see significant regeneration, and this is after 8 weeks. So the foam, most of the foam it should be gone, right? And then this is a new tissue that's being formed. But then on the other hand, if you look at the VML untreated, still you see, basically there's a significant deficit, right? So then we looked into the functional recovery, and we noticed that there is a significant functional recovery in both cases. But then obviously when we had IGF, it was more pronounced. But one of the interesting points for us was to look into how this thing is actually, or how foam can be compared to regular hydrogel. And we did a subcutaneous implantation of the material in rats, and if we implanted foams, we implanted the gels. And then when we looked into that after, I think it's after 7 days, you can see that, first of all, the foam is barely visible. There is no clear cut, kind of border from the foam and the tissue. So it's really realistically integrated. And one of the other interesting things that we observed was that in the foam, we had a lot of new vessels grown, and then the vessels were carrying, you could see even RBCs in those blood vessels. But on the other hand, in the gel, when we looked into that, after 7 days, still you don't see much of selling growth, right? And even the border is not properly, is still, kind of properly distinct, right? So you don't see much of it integration. So that was actually supportive of, or suggesting that our hypothesis that the porosity or pore size distribution is important was true. So then we went back to our VML injury model, and we did some histological analysis. Initially, we did trichrome staining, and we observed significant fibrosis in VML injury, which was not treated. And then we saw basically less fibrosis in the group that received foam, and also the same story for the group that received foam and IGF. So the other interesting observation was that we had basically new fibers being formed. So then we also had the way that we identify new fibers was that usually when you have new muscle fibers, the nuclei is at the center, and then as they mature, the nuclei move to the periphery. So then in the group that was injured and then received foam, we had a lot of nuclei that a lot of muscle fibers at the nuclei was at the centers. So this is still ongoing. We have also seen that these scaffolds are basically significantly contributing to even functional recovery combined with basically scaffolds combined with physical therapy. So that's an ongoing research, and we're hoping to publish another paper recently, which is shining some light on the combination of regenerative medicine or kind of scaffold therapies versus physical therapies. So I should mention that I do have conflict of interest, or kind of some financial interest in a company that's called Imprint Bio, and they are trying to translate this technology into clinical practice right now. The other thing that one of the other things that I wanted to discuss today was basically our research, or some of the research aspects that we are looking into regular bioprinting process, right? So the most common bioprinting technology or bioprinting strategy that's being used is extrusion-based bioprinters, right? So one of the issues with those bioprinters is that typically the size of the filaments or fibers that you generate are, few hundred micrometers and above, right? There are, some of them that possess better resolution, but most of the common ones, the resolution is not as good as, less than a couple hundred micrometers. But then when you think about it, when you read the literature suggests that if you want the topography of the fibers have immediate effect on the cells, usually you should go, limit dimensions to about 100 or maximum 200 micrometers, right? So then this means that, if you print larger structures, larger fibers, the cells are not going to follow the topography easily. So one idea that we had was to, somehow generate topographies or generate intrinsic topographies inside the fibers and try to, kind of increase or improve the resolution, or at least topography of the fibers that are being printed or bioprinted using regular bioprinters, or low-end bioprinters. And for doing that, we utilized basically a static mixer in a way that you can mix materials together. So in this case, the green color that's shown on C is a non-cell permissive component, which was alginate in this case, and red was basically gelatin metacolloid. And then that compartment was carrying the cells. So then cells, the hypothesis was that cells would sense the topography of the generated by alginate layers, and then they would orient themselves along the lengths of the fibers. And this figure is actually showing or confirming that, basically when we had the topographies, within a day we had, in 2 days, sorry, we had basically very good alignment in the fibers in comparison to the other one, which we barely have good alignment in the system. So then this is actually a strategy that can be used both in 3D printing and also in biotextiles. So then you can even imagine generating larger fibers and then assemble them using different textile processes. And then, as you see, still even these fibers are fairly large. You can look into the organization. The cells are following the topography of the fibers very, very well. So the other, I'm going to switch gear and move towards our research on chronic wounds. So again, the skin is another exciting tissue which possesses excellent regenerative capacity. But despite this interesting regenerative capacity of skin, there are 6.5 million Americans that are suffering from some sort of chronic wounds or burns, right? And then when you think about the basically the cost that is being exerted on the healthcare system right now is about $25 billion, which is expected to be doubled very soon, right? And then these chronic wounds are the leading cause of limb amputation. They can affect the patient's quality of life significantly. But the issue typically is multiple factors, right? But if you look into the wound healing cascade, so initially when you have an injury, the bleeding is going to be stopped, and then once the bleeding stops, then you're going to have inflammation. Basically, immune cells get to the site. They try to remove debris, to try to clean up the areas, remove pathogens, and then they start triggering proliferation. And then vascularization is going to happen. Fibroblasts are going to deposit extracellular matrix. And then as it progresses, cells start remodeling, and then basically cells that are not needed anymore are going to go through apoptosis, or some of them are going to be removed by other cells, and then eventually you're going to have healed tissue. But it's not always the case, right? Especially if the patient is suffering from underlying conditions, or the injury is massive or very deep. So those things, this orchestrated cascade of events can be interrupted. So then in those cases, usually the wound is going to be locked in an inflammatory phase, right? Or sometimes even called hyperinflammatory phase, right? So the challenges that are preventing wound healing are typically lack of angiogenesis as vascularization, and this is very common in patients with diabetes and obesity, and then hyperinflammation. People with diabetes, they do have some level of chronic inflammation, and then if there is an injury, that inflammation can be magnified. And the interesting point about inflammation is that you need inflammation, but you don't need too much of inflammation, right? Because the inflammatory cells are going to secrete cells that secrete factors, or secrete cytokines or enzymes that are responsible for removing extracellular matrix. And if you have too much of that, then any extracellular matrix that's being deposited by other cells are going to be removed. So then there's no regeneration. And then obviously when you have an environment which is hypoxic, rich with nutrients, inflammatory factors, it's perfect for pathogens, right? So then when pathogens get to the injury site, they basically quickly form biofilms, and then they try to hijack the immune system and then quickly things can be life-threatening. But then when you look into the clinical practice, the state of the art of clinical practice is shown here. We do have, some skin grafts and some, scaffolding materials that are being used. Most of them do not release biological factors or any drugs, but there are maybe a few that does that, but still monotherapies, right? And then there are some mechanical tools which or physical tools, because some of them are mechanical, some of them are optical, basically electrical, and those are the tools that are being used in clinic, right? But then when you think about the drug delivery or growth factor delivery for wound healing, everyone has focused on what and when. And the reason is that, first of all, you need to know what you are delivering, right? And then, as you see, there is a cascade of events here, right? So then you want to deliver the right therapeutics at the right time, right? So then when is becoming an important factor, and people recognize that, right? But then the question is that how to achieve this in clinical practice, right? Because when you talk to clinicians, a lot of times they don't have much information available to them, right? And if there are dressings that can provide information to care providers, that's going to help them with decision making and designing the treatment design for a specific patient, right? So one of the projects that we started working on back in 2014 was a project that was a collaboration between our medical school, Tufts, and Purdue, and the idea was to design basically an automated smart dressing in a way that it can sense the environment, and based on the sensing values or the data that was generated by the sensors, it could interpret the situation and the condition of the wound and then release therapeutics as needed. Right? So obviously it was a big goal. So the very first version that we started to, or we focused on, was a simplified version which had pH sensors and temperature sensors, and then pH was considered to be kind of an indicative or a marker of infection, and then it was it's in the literature it's reported that most of chronic wounds their pH is either significantly elevated or significantly lowered, and that's related to infection. So then we thought, if we have pH sensors that could monitor the environment, and then we have drug carriers that could release antibiotics on demand, if the pH goes significantly high or down, then that's the time that we can assume that's infected, and then we can release antibiotics. So I'm not going to talk about the sensor designs and the electronic designs, because it was done by our collaborator. I'm going to focus mostly on the drug delivery aspect of the system. So for drug delivery system, we looked into thermoresponsive drug carriers, and the reason that we looked into thermoresponsive systems was that temperature is proven not to be harmful for tissue or tissue regeneration, especially if the temperature is at moderate levels, right? Obviously, if you burn tissue, that's a different story, right? But then we looked into NIPAM-based polymers, and NIPAM-based materials are known to basically translate from a hydrophilic state to a hydrophobic state, and the critical temperature is about 32. So we generated relatively larger particles to be able to have multiple rounds of release from this system. And as you see on the second row, you see the mechanism of action, right? So the molecule is entrapped, and then as basically you heat up the system, the particles collapse, they release their payload, and the payload is being diffuse, is going to diffuse into the environment, right? So then we encapsulate these particles into an alginate gel, which is basically an FDA approved material for wound healing, and then we looked into the release kinetic. But although we had relatively controlled, we could control the release rate by adjusting the temperature, but still we had a lot of unwanted release. But the other thing that we noticed was that even when we we were turning on the heater on and off, we had, basically we could adjust the temperature, the release, or we could, achieve multiple rounds of the release, but the amount of the release was inconsistent. But then we went basically generate a variable integrated patch, which was basically a heater basically casted directly on one side of the hydrogel patch, and then sensors were interfacing with the tissue. Sensors were measuring the signal, the pH of the environment, and then the signal was being processed on board. And then once the controller was detecting, basically a critical pH, then the heater was being activated. The heater was heating up the hydrogel patch, and the drug was being released. So we looked into the potency of the antibiotics that were encapsulated, and we observed, the potency by a zone of inhibition assay, and later on by a CFU count assay, and it was suggesting that the antibiotics are potent, and only antibiotics are contributing to the eradication of the bacteria. We did some in vitro experiments in which we infected culture of basically endothelial cells, and in the positive control we added antibiotics to the culture, and we had a group which only had alginate, no particles and no drugs. And then as you see, still the scratches say the scratches remained open. But then we had another group which had all the components of the system, and then as you see, antibiotic is being released from the system, and then you see scratches being closed, and the pathogens are not interfering with the scratches. Then we designed an in vitro model which was kind of mimicking the environment of the wound. So what we did, we had a chamber which we inoculated bacteria in there. It was the chamber was continuously perfused with a cell culture with the pH of 7.4, and as you see, initially the pH was detected at 7.4, and then as time passes, the pathogens are basically or the bacteria are growing, and then they change the pH of the chamber. And we defined the pH, the critical pH for the device to be 6.5, and as it hit the pH of 6.5, we activate, the heater was activated automatically. And then in one group that antibiotic was loaded on the system, you see that quickly pH is recovering. It means that the bacteria are dying, and then we have continuous perfusion, so that the pH is slowly recovering. But the other group, the pH is not recovering, and pathogens are there. So, but again, as we discussed, most of the times we are dealing with multiple challenges which are leading to formation of or occurrence of chronic wounds. And then this means that you're required to be able to release multiple drugs. But then one question that we had, how can we generate systems that we can release multiple drugs with independent dosages, etc.? So we decided to weave a dressing in which that every single thread is basically a heater and loaded by drug carriers that are carrying a specific dose of a particular drug. So in that case, we could basically pack, multiple drugs, or multiple doses of a drug or different drugs on a single patch. And then once one of the threads would be activated, only that particular thread would release the drug. And every single thread was addressed by a controller that was on the system. And the other thing that we did to be able to better control the release rate and the critical temperature, we created basically copolymers of NIPAM and PEGDA. So then in that case, as you see here, we don't have much of unwanted release at the room temperature, so which was very important. And then we looked into the activation of the system and under IR camera, and you see when the two threads are being released or being triggered, only those regions are basically heated up. The rest of the patch is not heated up, as you can see. So then we went, did the same story, looking into the potency of the drug that the antibiotic that was being released. We did zone of inhibition test, and also we did CFU count test. And in both cases, the antibiotic was potent. But then we decided to create basically. I'm actually checking my time, so I already talked too much. So I tried to finish it in 5 minutes, 10 minutes. We tried to create an in vitro model.
We should be okay, Ali. We should be okay.
That's great. So we created 3D culture of HUVEX in the endothelial cells, and then we contaminated this culture by staph aureus, and then we placed a patch on top of that system, and the patch in one group didn't have any drugs. In one group, group F had basically antibiotics loaded, and in group G it had antibiotics and VEGF, which is an angiogenic factor which is supporting vascularization in endothelial cells. And one of the things that we observed was that in group E, as you see, pathogens are slowly growing, and then they change the environment, and then this is basically a live assay, and live cells are appearing in green, and then dead cells are appearing in red. And as you see in E, at the end of the day, we have many dead cells, right? But in F, we had antibiotics being released, so cells are surviving, but we also don't see much of vascularization, right? The endothelial cells are growing, but only few, capillary-like structures can be seen. But in group G, which antibiotic was released, so cells survived. But then VEGF was being released, and you see that significant vascularization in the cells that are receiving, basically VEGF at the same time. So it was very exciting. We decided to take this into and test on animals, and we created basically animal full thickness injury wounds on diabetic animals, and we placed the patch on which had VEGF on that, and then we released VEGF through the patch. And after 10 days, we went back in and we tried to look into what's going on. Over time, we saw a little bit of reduction in terms of the wound size, but we saw more significant enhancement as on the thickness of the granulation tissue. But still, the difference was not very significant, right? This is VEGF, which is an important angiogenic factor, and that's what body uses for starting vascularization post-injury. And if this doesn't have much of effect, that was strange and interesting to us. And the question for us was that what's going on? Why we don't see significant effect, right? Then we did I did more literature survey, and we noticed that VEGF has also failed in clinical trials, although it was successful in vitro, in vitro experiments, and also in some animal models, right? So then I started, asking questions. I started putting my mechanical engineering hat on, and I noticed that, well, most of the cases, wound is covered by a layer of necrotic tissue, or there's a crust, etc. And even if you debride it, still there are some cells that are not very active, right? So then the cells that you're targeting are under that layer. Then these cells, most of chronic wounds, are exuding. So then there is convective flow from inner layers of the tissue to the towards the outer layers, right? So then you want to diffuse, or you have your molecules diffuse inward, right? And these are large molecules, right? And then exudates typically rich with enzymes and pro-inflammatory cytokines, etc., which is going to deactivate or denature those proteins, right? So then you're not going to have a significant local bioavailability of the drug. So then we tried to, and also the other issue that still we had was that, although with all the basic modifications on the material that we did, if a patient was wearing this device on a hot day of summer in Texas, everything would be released at the same time, right? So then we tried to go back to make things more reliable in a way that, if at some point becoming someone wants to use it in clinical practice, it can be precisely controlled, right? So then we went back to regular mechanical pumping systems, but then to be able to kind of reduce the traveling distance of molecules, we tried to utilize macroneedle arrays, and basically to penetrate the tissue and release their payload, or the drug into the tissue. So at the beginning, everyone was surprised that we want to create a secondary injury in an already injured tissue. But we thought, maybe, the outcome is going to be, at the end, is going to be better, right? So then, for making the needles, we decided to use 3D printers, and 3D printers, multi-material 3D printers, enabled us to make needles which were rigid, but the backing was flexible, so they could conform to the tissue, etc. And then the needles, we tested the needles were not breaking, and even if you would compress them significantly, they would bend. Right? So still, they would, if you want to pull them off, you could pull them off, and then they could easily penetrate the tissue. So the sharpness was in good shape. Then we designed our electronic module, which was basically the part that was more expensive, the other part that was disposable and could be replaced. And then we utilized 2 miniaturized micropumps, and then these pumps showed that they enabled us to control the release rate precisely. So we could control the flow rate by adjusting the applied voltage and also turning the voltage on and off. So then technically we could deliver any amounts that or any dosing that we wanted, right? So then we looked into the release kinetics or assessed the drug distribution, and we created an in vitro model that was mimicking the environment or the architecture of chronic wounds. So then we utilized cell culture inserts. We add a layer of agarose to mimic that necrotic tissue, and then we either applied the drug topically or on top of that, or placed our needles into the gel and then released the drug in there. And then we didn't poke the membrane, so this schematic is incorrect. But then at the end, when we sampled the bottom chamber, which was technically mimicking the environment of cells that we are targeting, we saw significantly more protein down here, which was very important for us. But then we did some animal studies. I'm going to go to those to discuss them, but the results were interesting. But then we decided to ask another question, right? Is the transdermal delivery only important? I'm not sure if this is the correct terminology, by the way, because there's no dermis here, right? It's a full thickness injury, right? Or it's if the depth and architecture or point of delivery is also important. So then we try to utilize another tool that have been used, which is liquid jet injector, which is utilized for delivery of insulin to deeper layer of the tissue or muscle. But generally speaking, it has utilizes high velocity jet, opens the pore in the skin and tissue and send the drug in deeper layers. And we compared the distribution of the drug in an agarose sheet. And this is the macroneedle array. We have a better distribution or deeper distribution in comparison to the topical application of the molecule. But still the gradient is from inner layer towards the outer layer. So the highest concentration is at the border, right? But then with liquid jet injector, we had the drug was penetrating significantly deep, but the gradient was somehow most of the environment or most of the cases inversely, because the majority of the drug was packed at the lower portion or locations in tissue, right, or the deeper location in the tissue. So then we try to, by this background, we try to look into the animals, the response of animals to the system, right? Having said that, liquid jet injectors, testing them on mice was a nightmare. We ended up killing many mice to be able to optimize how much of pressure we can apply to safely deliver therapeutics. But eventually, what we noticed was that there are a few things that I want to point. Negative controls or delivery of PBS or vehicles, etc., didn't have any effect, and even topical delivery of VEGF was not significantly different from them. So no treatment with VEGF was very comparable to even treating the wounds with VEGF topically, right? So that was exactly what people have observed or used to observe in clinical trials. But when we delivered VEGF via liquid jet injector and macroneedle, we saw a little bit better, at least for liquid jet injector, but not significant, but it was a little bit better. But with macroneedles, it was significantly better. So when we histologically looked into the tissues, we observed, basically more advanced wound healing in the group that received VEGF through microneedles, and also we had more established blood vessels in the group that received VEGF through microneedles. Then we went and performed another study on pigs, and this was acute injury in pigs. So these pigs were healthy, so we know that they're going to heal. But when we looked into, we tattooed the border of the bones, and one of the things that we observed was that so let me also say something else. So because pigs are the wound healing, the healing mechanism in pigs is very similar to humans. So they in pigs there are two mechanisms: reepithelialization and contraction contribute to healing. So contraction is more of a reparative mechanism. Regeneration is basically, or epithelialization is the regenerative mechanism. So contraction helps with the closure, but significant contraction means that you are changing the properties of the tissue, and that can lead to significant scarring and re-rupture of the tissue, right? So then one of the things that we noticed was that when we delivered VEGF via microneedles, it actually significantly reduced the contraction. And then when we did histological analysis, we observed that basically, again, more blood vessels, more mature blood vessels, less inflammation, etc. And then we are basically working on making other types of hydrogels which can control release drugs. We're also looking into different types of therapeutics, especially those that are contributing to, or they can modulate immune response. And we have seen very interesting results, and we are hoping to publish them very soon. So the last thing that I want to talk about is that not all the times, microneedles are very exciting, especially when you are dealing with superficial wounds, right? But when it gets to deep cut or kind of gaping wounds, probably microneedles are not going to be, or those factors on its own are not going to be sufficient. So you need to generate some scaffolding materials to be able to support the regeneration, right? So then we try to again utilize our famous GelMA mixed with VEGF based on the results that we observed in our animal studies, and look into if it's actually supporting tissue regeneration. And again, we used our pen printer to in situ deliver the material, in situ cross-link the material, and because there was no need for suturing, the material was self-adhering, and everything was good. So then we went back and looked into the pigs, and then the response of the pigs. So the animals that received the scaffold in general had less contraction in comparison to the animals that they didn't receive scaffolds. And the other thing that we observed was that when we looked into the histological analysis, if you compare these, these four kind of figures together, micrographs together, you see there is a significant difference in the number of red ridges in the groups, right? And the group that received the scaffolds with VEGF were far more advanced in terms of healing, right? We were actually approaching the original tissue. When we characterized the reepithelialization and we characterized the scar index, it was suggesting that the delivery of the scaffolds with growth factor or with VEGF significantly improved the reepithelialization. And then at the same time, when we had the scaffolds carrying VEGF in situ print on the wounds, they actually significantly lowered the amount of scarring, right? Which was very interesting. So, again, this is a very robust technology that can easily be translated into clinical practice. And again, this is one of the areas that Imprint Bio is looking into, translating it into clinical practice. So with that, I would like to thank my team. This is, basically the first picture is actually when we were back in Nebraska, and then this, the one on the left side is actually a more recent one, which was taken in at UConn, and we managed to take our masks off for a few seconds, just take the picture. So with that, I would like to thank you guys, and thank the organizers. I will be happy to answer any questions.
Thank you very much, Dr. Tamayol, for your very interesting and inspiring presentation. So I'd like to encourage the participants to ask their questions using the Ask a Question box, which is at the bottom of your screen. I would like to also remind everyone that our next speaker is Professor Dino Di Carlo from UCLA. Next week, make sure that you follow us on Twitter at TransLBME to get most up-to-date information about the upcoming talks. Well, while we're waiting for the participants to ask questions, I have a few questions myself from Ali. Again,
maybe Ali can stop sharing.
Ali, you can stop sharing. I mean, if you,
so he can see us.
Right, that's much easier. It was
so Ali, a very interesting work on using biopens or this basically 3D printing pen for musculoskeletal tissue engineering. So since this is the theme of these series is on translational biomedical engineering, and we typically talk about some of the challenges in terms of translating this technology into the clinic, we would like to know your thoughts on these challenges and translational challenges of the technology that you've developed, and it could be from the bioink design and then also the biopen itself.
So I think, thank you for this important question. I think I want to actually step a little bit back, right? When we started this project, it was with a close collaboration with Dr. Indranil Sinha, who's a plastic surgeon at Brigham Women's Hospital. He has interest in treatment of volumetric muscle loss. So one of the things that has, kind of helped us to get to this point was the continuous interaction with him and other clinicians. So we came up with different strategies, and each time we, ran them by them, right? They raised some concerns, they raised some questions. And when I showed him this idea and showed him this device, he mentioned that I can't see myself using this in a surgical room, right? So then that was very helpful. Then later on, he even tested the system. He gave us some feedback on how to design, how to modify the design in a way that this is something that they can use it in during the surgery, right? So that's one aspect, which is getting into the acceptance and also understanding your customers, right? So to be able to generate something that they can use and they like to use, right? So that's one part. The other part is going to be, basically when it gets to translational research is interaction with the FDA, right? And regulatory process, right? So we have been working on months, and then slowly we get to the point that a lot of times, you shouldn't. Again, getting back to the point that engineers try to overcomplicate things. So when it gets to translational research, you should go with absolutely minimum possible complication, right? The simplest thing that can get the job done, right? So that makes your life much easier, right? And try to ensure, that the regulatory process, try to stay on the device side rather than, biologic side or combination side device. So that those things makes your life easier. But then another side of the story is the commercial aspects, right? Because you need to be able to raise funds, and you need to eventually be able to, sell your product and be successful, right? And then you should understand your market, right? What is your niche area, right? What is the area that you want to cover? What type of injury you want to cover? Are you going to go after, big injuries? Are you going to go after smaller injuries, right? So to me, that's also an important factor, which we spent several months trying to interview different plastic surgeons who are active in this space to figure out what type of application we should target at the beginning.
Okay, very interesting points, and I hope that the other participants actually appreciate these thoughts. It's very important to when you design a new project or you design a new product, you think about these aspects, including the usability, the regulatory aspects, and then also the market aspects during the design. So integrate all of these aspects during the design of the project. And I think the best way to address these aspects is to start the conversation with the end users right from the beginning of the project. Thank you very much. So I'll get into two more technical questions now that we talked about the translational aspects of these technologies. So my next question is on that foam-based bioink that you folks developed, and then I wanted to hear your thoughts or to hear your comments about the ability to control the pore size in these foams. How much you can control the pore size, the uniformity, and etc. If you can comment on that, that would be great.
I think that's actually a good question, right? So again, it depends on what strategy you use to generate those foams, right? So the size of these pores is an interaction between the shear stress that you generate or apply on the air pockets and how far you can break them down, right? So then we looked into different scenarios or different process parameters. So the agitation speed, the protein concentration, the basically the protein charges, and the use of basically emulsifiers are the factors that enable you to control the process, right? We also did some studies on exactly making canister-like systems in a way that we can deliver them like shaving foams. So with that also we could generate foams, and in those cases the amount of pressure that you apply is affecting the portion. But the uniformity is an important factor that you raised, right? You want to ensure that the pore size are uniformly distributed, and if you agitate them long enough and uniformly enough, you can generate basically uniformly sized pores. But then we are also looking into other technologies right now to try to kind of utilize some conventional fluid mechanics systems to be able to generate pores or foams with pre-designed pore sizes. Because in these cases, we process whatever happens, right? Then you can see the trend. But if you design it and you generate the pores yourself, so then you have control over the sizes.
That's very interesting. Again, it's very cool and interesting to see that these technologies use engineering tools to design new materials, which is.
I spend a lot of time on eBay. I spend a lot of time on YouTube watching cooking movies or cooking.
Well, yeah, that's really good. That's really great, and then that's how these new technologies are being developed. So I have another question about now your wound dressings technology, and then especially I'd like to ask about the dressings that deliver VEGF. And my question is about the stability of VEGF. So how stable it is especially in terms of temperature, when you apply those temperatures. Maybe if you can comment on the temperatures that you reach using these heaters, and then how much you think it would affect the stability of VEGF.
I think that's actually a good question. So any protein becomes more unstable as you heat them up, right? So then, but we try to keep the temperatures below 40 when we're activating the system. So then, to avoid kind of significant tearing of the protein because of the heating, right? But then VEGF relatively is more stable than some other factors. So the literature suggests that it's stable for a couple of days. In vitro, in incubator space, and even in vivo. So we assume that it should be we made our assumption the same way. And when in the animal studies that we did, I forgot to mention, we did rounds of the VEGF, and each round was separated by 2 days. So assuming that the VEGF is going to be stable for 2 days, and then after 2 days we renewed VEGF in the animals.
Okay, so then
something else that I should say is that this is because you are it's like a bolus injection, right? So the VEGF is not encapsulated in anything to protect it, right? So if you can encapsulate it in materials, then the activity will be longer.
Okay, longer. That makes sense. So then, well, my last question would be about that pump-based microneedle technology, which is really cool. But I wanted to know how you envision this device to be used. Is it going to be similar to those maybe microneedle-based insulin pumps, something that is
Right.
but Medtronic? Is it something that you have in mind? Maybe if you can elaborate more.
So I think that's actually a good question, right? So then I think the concept is kind of similar, right? Because it's an infusing device, and you can have it like those insulin delivery platforms, right? But then the key question becomes how you want to market this, right? Do you want to market it as let's say an infusion device, which the care providers are going to load any drugs that they want into that and deliver that? That's one strategy. Or you want to sell this device with a drug, right, a specific drug, and say that this is the drug goes into the device and you are going to deliver this, right? So that's something that we are not still sure about, and we are debating it. But obviously, if you go after the first route, it's going to be easier, right? But the key thing is that there is no gold standard drug for wound care, right? So although it's a very well researched area, but there is little established in terms of treatment of wounds, right? So that's the challenge that we are facing. But for example, you mentioned for insulin delivery, that's insulin is the drug, right? You know what you're delivering, so then you can easily say that it's going to be an infusion device, right? We are not going to make any changes to the molecule, right?
Cool. So I promised to ask the last question, but I have another question now. Do you think, and this is a hypothetical question, do you think these microneedles can be used to inject stem cells deep into the wound to promote wound healing?
That's actually a good question, right? So I think we have done it before, right? And it can be done. The issue is not delivery of stem cells. The issue is more protecting the stem cells after delivery to ensure that they stay in the site and they're not doing anything strange, right? So Ali had a paper that they used microneedles for delivery of the cells, but I don't know how successful was that in terms of keeping the cells alive, etc. Right? But at least they saw some outcome improvement, right? That's something that we are trying to do. We're also looking into some new technologies right now that significantly improve our ability to deliver the materials or microneedles stem cells in the form of microneedles directly into the tissue. Okay. But that's actually a very good point, and I think that is going to be very important if you can utilize patients' own cells for treatment of that's going to be significant.
Awesome. So I think if you let me, I keep asking questions, and I know that our speaker has a very busy schedule. So if the audience doesn't have any question from our speaker, I'd like to thank our speaker for his inspiring talk. Very interesting as usual. and it's great to have had you with us, Ali. Today, I would also like to again remind everyone that our next speaker is Professor Dino Di Carlo from UCLA. Make sure that you mark your calendars for next week. And then with that, I'd like to conclude this session and then wish everyone a wonderful day or night ahead of you.
Thank you, Mohsen. It was a pleasure, and again, thank you for the opportunity.
Sure. Take care. Have a good day.
Have a good one.